Hello and welcome to WX15, pronounced Wex. I'm gonna play this game for Double Feature Friday called Mutation Madness. I have no idea. It's just DNA scrolling across the screen. This title screen's not very impressive. So let's jump in. The epic story. You are a genetics specialist getting tested. You have decided to get tested because your father has recently died of a disease that is caused by genetic mutation, and the mutation is something that can be hereditary, which means it can be passed down from your parents. I love my features. Am I standing or sitting? The doctor's ready to see me, and I teleport. Okay. Well, double doors and. Okay. You are here to get tested for a genetic disorder that can be passed down from your parents. Different traits are passed down from your parents to make you who you are. In your body, DNA has to copy itself in order to make more cells, to help you grow and stay well. While this is happening, something can go wrong. This is a mutation. So I'm Wolverine. The DNA mutation minigame. This part of the game will help show you how DNA replicates and an example of what can go wrong. DNA replicates itself through the nucleus of a cell before a cell goes through mitosis to create more cells. During the replication process, mutations can happen when something goes wrong. If it doesn't get fixed, the future cells that are made off of the mutated one can have the mutation as well, which is actually survival of the fittest. Those with the mutations that work that help the animal to survive better, and then mate, and those mutations actually become normal. Um, you will see the DNA double helix untwist, and different nucleotides match up. They know where to go because adenine always matches up to thymine, and guanine always matches up to cytosine. If I pronounce those wrong, kill me in the comment section. If you mess up in matching, you will create a mutation in the DNA. Double helix unwinds. Colors. As you saw, the nucleotides were being matched up with the DNA. The green would always match up with the blue, and the red would always match up with the yellow. This is true with the DNA. The green represented guanine, and the blue represented cytosine. The red represented adenine, and the yellow represented thymine. Now the next one you choose. With the next one, you will choose which nucleotide will go next. Blue and green, yellow red, red yellow, green blue, yellow red. I think guanine. You have matched nucleotides correctly. Click here to continue. As you can see, a mutation can be created very easily, but we didn't make a mutation. Transcription has a very similar process. Okay. Transcription is a similar process, but it is meant to make mRNA. mRNA is a very similar copy of DNA, with the difference being that instead of having thymine, it has uracil. DNA is deoxyribonucleic acid. mRNA is ribonucleic acid. Cells make mRNA because it is too risky to send DNA out of the safety of the nucleus. The mRNA is used as a set of instructions to make proteins in the body. Mutations can occur while the mRNA is being made. DNA, RNA. Okay. Uracil goes with the adenine. Okay. Red. Yeah. Blue. Well, where'd that pink one? Okay. I'm gonna say pink. You have made a strand of RNA that is ready to exit the nucleus and go through the process of translation to make proteins. Translation separates the nucleotides into words that consits. So they're con men that sit. These con men sit down of three different letters and translate the word into what protein it should be. Protein's misspelled. Translation separates nucleotides into words. We said that already. Go make some alanine. No, you go make some alanine. I'm watching. We're going to perform PCR. I don't know what that is. to help study to see if you have the mutated gene. This will involve taking a sample of your DNA to be tested. We'll then make many copies of it by putting it in a solution of different nucleotides. And am I bald? I guess not. I got that little hair right there. Primers, which will attach to the part of the DNA that we want to see so that it will be the part that gets copied. Okay. That will then be put into a thermocycler. This will cycle different temperatures because the DNA does different things at different temperatures. The process of DNA duplication will then happen and we will be able to easily see if you have the mutation. I didn't even get to do anything? That's no lame. Thermocycler. Box. The primer is attached to the part of the DNA that is wanted to be copied. And that section of DNA will be reproduced many times. The DNA is then placed on a blue gel, just a blue box apparently, and the blue gel is placed in an electric chamber where the DNA is moved because of the electric current. Actual test, control, I don't, what? DNA samples at different spots showing whether it was positive or negative result. No, I don't see that. According to our test, you do not have the mutated gene. So this was made by students. Well, you just made it onto YouTube. Thank you. I feel that was rather short. I may do a third video for Double Feature Friday, and this will be going up soon. This is Black15 signing off. You're a wonderful audience. Click like, subscribe, leave a comment. Stay fancy.